coming to the next speaker. We're out of time. Thank you, Jeshi. It's a great pleasure to introduce Professor Dr. Claus Michael Lehr. He's head of the Department of Drug Delivery at the Helmholtz Institute for Pharmaceutical Research at the Saarland, at the Helmholtz Center at the University of Saarbrücken. And he's speaking about nanomedicines for overcoming microbial barriers, host cell membrane, biofilms, and gram-negative bacterial envelope. Thank you.
Right, thank you. Do I get a pointer? And great, yes, great to be here. Great to be invited again at CLINAM. I hardly missed one, actually, and it's always very nice, and I really enjoy being here and meet the colleagues. Yeah, some of you know me as a professor of pharmacy at Saarland University, which I'm still, but about 6-7 years ago, we had the opportunity to join the Helmholtz, this was not what I wanted, the laser is here. The Helmholtz Center for Infectious Diseases, where in fact Eva Medina, who has been mentioned, she's a colleague of mine, been working, and to build up with them together a first institute in Germany dedicated to pharmaceutical sciences from this non-university research organization called Helmholtz, which is actually the biggest research organization in Germany. So our center is actually located in Braunschweig, and the institute is in Saarbrücken, very near to the French border and not too far away from Basel, so that I can get here easily by train. This is the new building that we just had opened in October 2015, and this is actually why we are working on infectious diseases. Some years ago, people thought there's no more need to do something, but then there was a big developing gap, and very few new drugs actually are coming up, but probably this is returning. What had happened over the years was, while some wise man said we could close the book of antibiotics, these bugs that turned out to be resistant came up. And they became more and more, and we read it from the press that there is a big threat on that. On the other side, the compounds that can be used to control these diseases, and actually even more the companies that are working on such drugs, they decreased. So clearly a need where the public sector should do something, and that's typically where in our country Germany takes activities. And when you look at recent data that have been predicted from UK reviews and studies, and also has been recently adopted by WHO, and then when you look in the year 2050, I might be lucky to celebrate my 90th birthday by that time. Perhaps there's still a CLINAM meeting, and we will see what's going to be, but it's being predicted that more people are going to die from infectious diseases than from cancer. So there is a clear need to do something there, and that's why I really enjoyed the opportunity to gear my research toward this direction. So essentially, I keep doing what I always have been doing. Look how we can improve the delivery of drugs across biological barriers that are always between the site of administration and the site of action. And I like actually to do that in a needle-free way. So that's why I had always a focus on oral delivery, on pulmonary delivery, eventually on putting things on the skin. And now, in addition to these epithelial barriers of the host, we have 3 barriers that are related to microorganisms and bacteria. And still, actually, our approach is strategically in the same way. We have let's say, deep trust, put it in this way, in the usefulness of working with human, cell and tissue-based models to rebuild these barriers so that we can see what happens, which has hopefully a better predictivity eventually for the clinical translation. The animal models have their place as well, but we also know their problems. And on the other side, we work on drug delivery technologies that do indeed also include nanotechnologies to actually achieve the improved delivery. For today's talk, I have decided to actually focus just on these 3 barriers that will be the host cell membrane, the biofilms, and at the end, I will just touch also on a topic that I think is going to be of increasing importance, that is the barrier around the bacteria themselves, especially in case of the gram-negative bacteria. So let's see how this goes. So the first story is actually related to intracellular bacteria. There are some of these bugs that you can get for free on airports, train stations, or whatsoever when you have poorly cooked food. They might not really kill you, but it can be extremely pleasant. And what they do is they hide inside epithelial cells that they know somehow to invade. And the problem is, how can you get them? And we actually had developed a project that we thought maybe we can bacteriomimetic carriers, trying to hit these bacteria, to strike them with their own weapons. So the problem, for instance, gentamicin, a drug of a class that we just heard about it, that's an extremely potent drug, just it has problems because of its polarity to get inside the cells. That's the interesting things, parasites, host cells, bacterial cells, but this is not simply working. What we could do, we could use a structure that Yersinia is using, for instance, to open the door for their own entry and going into the cells. Intuitively enough, this protein has been called invasin, and there is Petra Dersch, a colleague in our center, who is an expert in that. So we talked together and said, maybe you can actually, I told to Petra, can make some of these invasins, give that to us, and we put it on some Nano carriers. For ease of simplicity, we started with liposomes. I simplify that largely, but you can imagine that this can work in this way. And then the concept actually would be that we could, the gentamicin-loaded invasin functionalized nanocarrier, talk to some receptors, use the same route of entry, and then release the drug and eventually kill some of the bacteria, some located in vacuoles and others not in vacuoles. And actually, let's say this summarized the data that we had observed, why all the controls actually confirm what you can read in the textbooks, that gentamicin is not infective against intracellular bacteria. With this kind of invasin-modified nanocarriers, we get a significant killing, at least, that we did not all of them might also have to do with resistancy or something. That's a first step that we really can do this in such a way. At this moment, we are working on alternatives to these liposomes. Polymeric nanocarriers might be interesting, especially because you can make PLGA nanocarriers in a way that you can stretch them and do them in an aspherical way, and perhaps that has implications for an even better uptake. This is still ongoing. Adriana Costoli is about to defend her thesis. She calls it rice-like particles. There is another student, Sarah Menina. She's going to have a talk on this. She started on another drug. Colistin actually is a drug currently in the clinic, a little bit underused because of several problems. Also, no way to apply it orally. And because we could, so we thought we could be using colistin and incorporate it in such a system. Also, by exploring another invasive protein. So instead of this Yersinia invasin, we're currently working on the so-called extracellular adherence protein from Staphylococcus aureus. This is a collaboration with some colleagues in our center, so we can make liposomes for that, and in order, because they should be used for oral administration, we check their stability in some biorelevant GI media. That seems to work. We can functionalize them with this extracellular adhesion protein, and we can see indeed that there is some uptake of these functionalized carriers, in this case in HAP2 cells. That's another epithelial cell lines, and actually we can see that regarding the uptake, at least, the EAP seems to do quite a bit better than the invasin. You see that also from the FACS data, and actually this is where this story at this moment has arrived, and we'll see how this goes further on. A few more details, if you are interested, on Wednesday by Sarah herself. So let's move from GI infections to, I told you that I had interest in the respiratory tract and pulmonary delivery, to respiratory infections, where we have gram-negative and gram-positive bacteria. Unfortunately, it's a mistake. The assignment is by the other way around, but this doesn't matter. Along the respiratory epithelia, we have a different physiology, and we have to get there. This is in the first place a piece of aerosol science and technology, which I'm absolutely not going to ignore, just these technologies of putting aerosols in the deep respiratory tract, they are there. And this is more difficult than getting things in the GI tract, but in the same way, if you just have it in the luminal side of whatever GI or respiratory tract, you know that you don't have complete bioavailability. What counts is actually how do you get after landings across this barrier. And this barrier is actually changing in case of the respiratory tract along the physiology. So from the airway zones with mucus, we get deeper to the lungs where we have ultimately an extremely thin alveolar epithelium, no more mucus there, but surfactant being present. And these cells are actually where many infections are taking place and where also things have to be delivered. When you want to work with cell cultures of the respiratory tract, you must make sure that you have the right cell types. This was a primary culture of human alveolar epithelial cells, which we published first in 1999. And compared to cell lines, you see that these are actually leaky like a sieve. That means A549 cells allow you interesting study with individual cells. But when you want to work with an intact epithelial, you must make sure that they have tight intercellular junctions. Now, not too long ago, in fact last year, we published actually a first cell line. We called it HELVI because it's human alveolar lentivirus immortalized cells that, as you can see, has type 1 cell-like properties and indeed has, over several, still get you used to this, over several passages, a very high transepithelial resistance of 1500 and more ohms square centimeter. So that's an interesting tool, and sometimes we still do the primary culture when we take not only the epithelial cells, but actually take from, this is human tissue that we get on a regular basis, the macrophages we can reconstitute and have the macrophages and epithelial cells in a co-culture actually. Then it comes to the question, how do you study aerosols in such a scenario? Because you have to face an air interface and you cannot work with pipetting solutions, at less than you would destroy a lot of the effects. So we started with sprayer techniques. We adopted impinger and impactor systems. In the beginning, I must say, relative naive attempts, but somehow they got us further. We came up then in 2010 with the PADOC, which stands for Pharmaceutical Aerosol Diffusion Device on Cell Cultures, that received some attention in the area of alternatives to animal testing because it's also useful for toxicological studies. The most recent thing is the new development actually of this PADOC cell, which we together with a company, in vitro cell, they call it dry powder chamber, and this is now actually commercially available, and it was interesting that even a company in Perth in Australia bought recently one, so my student could travel there and get it installed and operational. So now it comes to the bacteria in this context, and the first example is actually that we wanted to see, can we perhaps incorporate again a known drug, clarithromycin, into nanoparticles in order to again improve the delivery against intracellular bacteria. So what we could do actually was making nanocapsules of clarithromycin either in PLGA or in chitosan-coated PLGA particles. You see here the characteristics of these systems. They work actually all quite well. You can make them. You can convince yourself that there is actually no uptake in epithelial cells. This was a bronchial epithelial cell line. You can see that you see an enhanced delivery across this epithelium, which could be useful, let's say, for getting more drug into the bloodstream with such an administration form. We could also see that there is no change on epithelial barrier function, so there is no damage affected to the cells, there is no cytotoxicity, the viability is unchanged, and the resistance of this epithelium is also not influenced. Now, what makes it interesting actually is when we take macrophages into the game, then we actually see that there is an uptake of these particles into macrophages, which is desired. And what you also can see actually, that there is a co-localization of these nanoparticles with the endolysosome and with the intracellular Staphylococcus aureus. So you could perhaps indeed reach this guy, and that's what these data point to. If you don't treat, you don't kill any bacteria. With the nano-encapsulated clarithromycin, you get a complete killing of the intracellular bacteria. With the plank of those, of course, you don't. And with the free drug, that is the interesting control, you don't kill these bacteria as well. So that's an interesting concept, actually, in doing so and improving the delivery. Okay, this was, let's say, what I had planned to talk to you about. And we have discussed now the issue of the host cell membrane. And the next one that I would like to talk about is the bacterial biofilm. That is a barrier, actually also a diffusion barrier created by the bacteria themselves, especially in the respiratory tract, where we have Pseudomonas aeruginosa infections, especially in cystic fibrosis patients. What these bacteria do, they talk to each other. They have a system that's called quorum sensing. That means, Latin, how many are we? If they decide we are enough, then they change their behavior from planktonic to making a biofilm. And how they do this, talking, is by producing so-called signaling molecules, quorum sensing molecules. And the concept of one of my colleagues is actually to find a new way of pathoblocking by disturbing this bacterial communication system. So you are not going to kill the bacteria, which would feel the bacteria unpleasant and force them to become resistant. You would rather like to try to discuss with them, so to speak, disturb their communication so they would be unable to build a biofilm and just do whatever they want but leave the patient alone. It's a little bit simplified, but this is in truth the concept of going away from classic antibiotics, which always, if you want to kill somebody, he finds a way to escape from that. How to do this? Well, actually, you find molecules that are perturbing this system of cellular communication. And this was one of the best molecules here that they could use. Unfortunately, like often in pharmaceutical sciences, interesting pharmacology, but very poor deliverability, very lipophilic, poor water solubility. So we thought maybe we can incorporate it in solid lipid nanoparticles, which we then eventually could aerosolize, apply to the patient, and they could deposit in the bronchial region, penetrate the biofilm, and deliver the payload in a controlled manner. At least at the level of this piocyanine assay, which is measuring the amount of this virulence factor that's responsible for the side effect or for the pathogenic effects, we saw for the same dose an about 7-fold reduction of this critical dose, 7-fold improvement of the delivery by working with these nanoparticles, which is, I think, also something that should encourage us to go further. What we now want to do this more systematically is to build actually on models that we can use for these nanocarriers and evaluate them. So actually the dream is to have a co-culture of epithelial cells, as I showed you, but on top growing bacterial biofilms that all on an in vitro system where we have the possibility to deposit aerosols. They would hopefully allow to penetrate into the biofilm and kill the bacteria before the bacteria kill the host cells, so that you can really do these studies. This is what we had to make bitter experiences. This is unfortunately not easy. Whatever we tried, bacteria were overgrowing the epithelial cells, killed it at all. We could not get it at work. So it took us a while, but slowly we are making progress. What we had to do, we actually had to add additional mucus to these cells, where we also found out that the mucus is a very important protective against the bacteria. Then the next thing is we found out it is not possible to grow the biofilms on top of these cells because cells have no chance to survive that period. If you grow the biofilm separately and then transfer it as a pre-grown biofilm on this culture, you can do that. And then finally, actually, you have that system as a result, and you can, let's say, do these experiments that you want to do it, and then you can see it. And the first data show that when we indeed treat these infected cells with drug-loaded nanoparticles on the left side, we kill, except for a few that are still remaining, all the bacteria, while if you enlarge there, if you do the non-treated, you see indeed here lots of bacteria sitting on this lung-like model, and this is what we are currently doing to further improve this system. So these models, they are nice, but unfortunately it's much easier to just take an animal or to take some cells, and this is not so trivial. That's what I wanted to say here. Okay, few minutes left, so let's face the so to speak, final frontier, perhaps, if we really want to go and hit sites of action inside bacteria, then we have to think the ATME concept actually further. Being in the plasma is maybe not enough, especially when we consider that bacterial envelopes are much more complicated, especially in case of gram-negatives, than our mammalian cell membrane in the intestine, in the lung, or whatever the place. We have this delicate structure of 3 layers, which you actually can see composed of an inner lipophilic membrane, a intermediate hydrophilic peptidoglycan wall, and then you have the outer layer, which actually consists of an outer and an inner leaflet, partially with active pumps, receptors, and all these things. So this is actually what we want to overcome, and in order to do that in a rational way, it would be perhaps useful to have an in vitro model to do that. So having this in a system that we know from cell cultures that others have tried for Pampa and other systems to model simple lipid compositions of the skin or the blood-brain barrier, could we start reassembling that by working first with the inner membrane, putting then the outer membrane, and finally also having this in between. So simply speaking, this is you take a sandwich, you first put some butter on it, then you put some jelly on it, and again you put in the 3rd layer some Nutella on top. So hydrophilic, lipophilic, lipophilic in this sandwich. And how you can do that, there are ways to use liposomes, not as a model, but just to create these membranes. You have to make sure that you really do achieve what you hope to achieve. So you have to imply microscopical and other physicochemical aspects. Then you have to ask the first question, is it useful? For instance, by just working with the inner membrane, we compared the permeability of some dyes and some beta blockers across the bacterial inner membrane model only. This is only the inner membrane with that of the mammalian comparator model, which is the same, just one lipid structure but different lipids. And you can already see that there are differences. Well, the reviewer said at that time, actually, we want to see some antibiotics and not beta blockers. And then they said, okay, we take some antibiotics. So you see the difference also for ciprofloxacin and minocycline. You see also that polymyxin B, which is known to destruct the bacterial membrane, is only destructing the bacterial model and not the other one. So encouraging us that we are in a sense on the right track. So one model. Now coming to this thing, I have to take it shorter here. You have to make in the first step the inner one. Then you have to put as a spacer the alginate gel, and then you have to deposit the inner and the outer leaflet on top again so that you have this triplicate structure of lipophilic, hydrophilic, and lipophilic membrane. You can again look that you see what you have. We're still working on this, quite interesting X-ray diffractive things where we're still waiting for better resolutions, but we keep going. And let's say the encouraging result here that we found by, again, looking at molecules of our colleagues who found, let's say, in this series of molecules, a relatively similar potency in vitro by checking how well would this key fit into the ISLOC, so by just looking for the affinity to the receptor. And they found it quite the same way. On the other side, when they looked at the effect in, some people call it cellulo, which actually should call it cellular, if my Latin is correct, but we call it in bacterio. So they have to see how effective they are in bacteria. And you see some of these are active and others have no activity. And we thought maybe that actually would be explained by the different permeability. When you look in these data as such, they are not immediately so evident, but when you actually do such a polar diagram, then you find all these active molecules above a certain threshold of permeability, while the non-active ones are below a certain level of permeability. So that is at most the beginning, by far not the end, but in a sense it encourages actually to carry on with that. The next steps will be that we're going to miniaturize that, coming from a 12 to 96 ml bottle, and in parallel we try also to go a little bit from this 100 micrometer thick layers to a little bit smaller ones, but there's still enough that I can hopefully talk at one of the other CLINAM meetings in the future. With that, I'd like to thank you for your attention. I'd like to thank to my team that chose us visiting a steelwork in Saarland, while we had to protect us by these aprons against the hot temperatures intrinsic to our classical industry of steel. This is a little, it's not a conflict of interest because Pharm Biotech is fully owned by Saarland University. This is our spin-off that I'm officially in charge. If you're interested to want to use these models, you can talk to us, and hopefully there's perhaps some opportunities to visit us at the Helmholtz Institute in Saarbrücken. Thanks a lot.
Thank you, Professor Lehr, for this vast amount of information at a fascinating, breathtaking pace. So I think there must be many questions. Let's see in the room. To build such microphysiological systems, of course, is very complex, and that could be one of the reasons that people are stunned by it and actually having difficulties to formulate some of the questions. But yes?
Thank you, gentlemen. So, excellent talk. So if you're going to use nanoparticles for inhaled delivery, what is going to be the approach? Because you cannot, aerosolize them in the usual manner.
Yeah.
So what is the intermediary step towards achieving that?
Right. The nanoparticles will display their advantages when they're down there and can talk to the cells, but you have to bring them there. There are two ways to do that. We pursue them both. One is you can, for instance, with liposomes, but also with polymeric nanoparticles, making an aqueous dispersion. And then you use a conventional jet nebulizers, and then you generate the particles of 2,000 nanometer, so 2 to 5 micron droplets. And you can imagine that in each single of these droplets, there's room for more than a couple of nanoparticles. That's one way. So by that, you can actually deposit nebulized nanoparticles deep down to the lung. The other way, if you, let's say, in the long run, want to develop a dry powder inhaler product, there are technologies, and there are many people, some people who worked on that. You can make microparticles out of nanoparticles. For instance, that you make them first and you apply then a spray drying process where you use a carrier like lactose to make microparticles which have the optimized aerodynamic properties which you need to deliver them down to the lung. Then when landed, the lactose melts away, it dissolves, and the nanoparticle is again there.
So how do you deal with the mucociliary escalator? Because now you deposit it in, say, region generation 10 or 8 to 10, and you have to get down to 18 or 19.
You have to indeed make the aerosol in such a way that you reach the deep lung and not just the airways. That is a question, again, of aerosol technology. And there is, let's say, according to some of these toxicological models, for for density 1 and 2 micron, a monodispersed aerosol actually will go for a large fraction into the deep lung. And there are examples for that you can do it. I mean, inhalation of drugs like insulin, it works. You must not go just to the upper airways. So relatively simple aerosols that we are using for asthma treatment or something like that, when salbutamol is deposited in the upper part of the airway, that's even better. But that's not this kind of aerosol technology that you need. You have to invest a little bit more technology and effort, but it's possible.
Yeah, certainly dosing and particle size distribution are very critical technical questions that have to be resolved. And of course, there are some standardized methods which are not as advanced, particularly with standard inhalers, that wouldn't be suitable. So I also appreciate your development in this area because there is certainly a need and a void of having better technology to be able to standardize and also understand how we dose.
As our current working hypothesis is that the nanoparticles have beneficial effect down in the deep lung, we did not want to go through all this development work first, which you would have to do, but let's say that's the advantage what the cell culture models allow you. You can put it in place, and then ask the question, is my hypothesis right or not? If we find it disproved, we don't have to do the development. If we find encouraging results, we'll go for it.
Very good strategy. Are there any other questions? Yes, please.
I could hear you.
Anyway, so the question is maybe a naive question. So to improve the efficacy of the particles against bacteria in the presence of epithelial cells, so you need to tune, I guess, the affinity of particles to bacteria and minimize the affinity to the epithelial cells and also minimize the uptake. Can you tune this and so that the particle target only bacteria and don't get it?
At this moment, actually, in my team, we have not been working on any nanocarriers that are really supposed to penetrate the bacterial cell wall. They bind. There are people who are doing that, quite nice work. Not ours, unfortunately, though there are ways to make, like, according to the concept of bioadhesive particles, you can design nanoparticles that really, you see that as a coating around bacteria. Mm-hmm. And you can do this by specific receptor interactions. Luckily, the bacterial cell wall has specific characteristics different enough from mammalian cell that seems to work.
Thank you.
We, I think, still have time for some more questions. Yes, please.
I had a question regarding your clarithromycin nanoformulation. You showed that the epithelial cells don't take up the particles, and initially, I mean, it's probably helpful to address the bacteria that are phagocytosed by the macrophages. But don't you usually, over time, also see an infection of the epithelium? So wouldn't you need to treat the epithelium as well, at least at a later point in the infection?
Yes, quite right. And actually, I wouldn't have minded if we had seen some uptake. Maybe that's what this is, where you need strategies like this EAP or the invasin to indeed get particles in non-professional phagocytotic cells. That is the point. And probably the fact that you have a small fraction, 10% could be enough, of these bacteria that could hide in epithelial cells and act as a reservoir. That could perhaps be the reason why we cannot hit them and why they come back and cause resistance and all these problems. I see your point.
Okay. One more question there.
When you do this interruption of quorum sensing, if you kill a bacteria, you put there some poison, the poison goes to the bacteria is dead, and the poison did its mission. But in case of interrupting quorum sensing, this material has to stay there for a long time. For how long time?
Well, that is, of course, let's say that you get off-target effects from drugs that act, sleeping pills that cause phocomelia in babies. That is a known problem in drug development. And luckily, we are now in the state where we are going to explore the safety. In the first pass, you look for the effect, and you must maintain the level long enough, either by repeated dosing or by, that's one of the ideas of creating a depot that penetrates in the biofilm, avoids, which is then an advantage of the presence of a biofilm, the otherwise rapid mucociliary clearance, and you can really make a depot there and provide a controlled release. That at least is the idea, but it is, of course, not so easily done than thought.
Thank you.
Yeah.
There's certainly a lot of appreciation for working with complex systems, and also once you interfere with the balance, you create a niche or a gap for something, you will have to deal with the problem. And I very much appreciate the approach of your research group to take that in consideration, because those are really critical questions that we have to deal with clinically, and certainly also the impact of adverse effects and events that we need to take in consideration. So thank you very much. If there are no further questions, then I see you in the next sessions.
Okay, thank you.
Professor Lehr.